Okay, so I want to finish this module by talking about sort of a newer, more modern branch or subdiscipline of biogeography called phylogeography. And we've sort of been talking about, we started off talking about center origin theory and pan-biogeography, and then we got into sort of vicariant biogeography and some of the nuances of that. And I want to sort of present phylogeography as sort of the next evolution of our understanding of how these things kind of work. And what I want to say, what's kind of unique about phylogeography is that it's a bridge between the traditionally separate branches of microevolution and macroevolution, or different areas of evolution. And so if we think of microevolution as sort of population genetics and looking at things like population size and structure and migration routes and things like that, basically what's going on at the population level. Macroevolution is sort of this historical biogeography thing, looking at large scale changes in broad taxonomic groups, looking at area relationships and vicariants, um, how vicariance and dispersal relate to one another, and the geography of sort of evolution. Phylogeography sort of sits right in between these two and allows us to kind of connect these two things in a little bit more concrete way than just sort of guessing or creating, sort of hypotheses that might fit. Phylogeography allows us to systematically and scientifically link some of these things happening at broader scales to things happening at finer scales. A great example of this is here we're seeing the phylogenetic tree for the mitochondrial DNA haplotypes of populations of the tailed tree frog and their distributions across the mountainous region in the Pacific Northwest. And what basically you have here, so for these tailed frogs, is you have these two separate clades. You have an inland clade, which the phylogeography is here, and you have a coastal clade here. And basically what this allows us to do is look at the diversity that's within these two clades, and we can see some really distinct differences. So in the coastal clade, you have these really older lineages occurring, along the coastal mountainous regions, and you have much younger lineages. You can see basically the shallowness of these branching indicates much younger diversity and splitting of these lineages in here. And basically this might actually represent speciation ongoing in real time among this inland clade compared to this coastal clade. So basically it's a way to link, stuff going on to the population level at what's going on broader evolutionary scales. Another example would be to use basically to compare phylogenetic reconstructions, or sorry, phylogeographic reconstructions to things like ecological niche modeling. And so here we have an example of overlap between ecological niche modeling and the phylogeographic reconstructions of refugia during the late Pleistocene for the western diamondback rattlesnake and the California gnatcatcher, respectively here. And basically, if we assume that the phylogeographic reconstruction is probably accurate, I mean, the ecological niche modeling is basically a projection based off of, the relationship between the species and its environment. And then we can look at changes to the environment in the past and sort of guess at where it may have been based on that relationship. The phylogeographic reconstruction allows us to compare what we know about that species in, more its genetic lineages and evolutionary history to those ecological niche models and see how accurate it is. And we can see for the western diamondback, it's not very accurate. So what you have here in these green regions are the ecological niche modeling projections. And in this outline is basically the phylogeographic reconstruction of the refugia. And remember, refugia are just like areas where these species existed in the past in sort of disjunct populations as as the environment changed. Like usually we're talking about this in context of like glaciations, like huge glaciers are moving in, and a refugia would be like a pocket where a species could exist outside of that, or like, you know, in contrast to the area that's glaciated. So it's probably an area that wasn't glaciated. I don't know specifically if that's what was going on here, but these are refugia for this species during the Pleistocene. So we know it's probably related to glaciation. But basically the point is it's not lining up super well for the rattlesnake, for the Western diamondback rattlesnake. It does line up pretty well, or at least better, for the California gnatcatcher. So it allows us to adjust our ecological niche modeling using this information. Similar thing sort of going on here where we can combine the phylogeography with the ecological niche modeling. And here it's done in the Brazilian Atlantic rainforest and looking at refugia of the rainforest. And basically they looked at these tree frog lineages and found that the lineages is kind of complex to look at this, but basically you had two areas of refugia, a southern area and this northern area here, and it's kind of in black and you can't really see it that well. But basically the lineages of these species lined up really well with the forest refugia in that you have these separate lineages, whether you were looking at a population from the southern part or from the northern part. So the southern part is in yellow here and has basically its own lineages, and then the northern part is sort of in the pink here and has its separate distinct lineages in these species. So it allows us to tease apart some things like that. Here we're looking at a mitochondrial gene tree for the red-tailed chipmunk. So this is, this is not phylogeographic. This is just sort of looking at the phylogeny, not the phylogeography. I realize it can get a little confusing with all these names, but this would be just the phylogeny of the species, and you can pretty clearly see an eastern clade and a western clade here. We're labeling these so that you can understand what's going on here, but this whole clade is from eastern populations, and this one is from western populations where they exist. But okay, so what we can see is like, okay, there's these two clades, but then there's also a lot of diversity within these clades as well, and diversification happening within these. So what does the phylogeography tell us? Well, it allows us to look at some more fine scale patterns here. And we can see basically, we can do this nested sort of phylogeography where you're seeing basically these branches represent mutations of the mitochondrial DNA that was sampled. And you can see, here's the western clade, and then you can see within the eastern clade, there's a lot of diversification, and we can map that out geographically to see sort of at different scales here. We're sort of like zoomed way out here, and here we're looking at some of the sampling distribution, and we can see their overlapping or non-overlapping geographic ranges. Basically, you have this western clade that's almost completely separate from most of the eastern clades here. So it allows us to get some more fine scale geographic and phylogenetic information about these species, or this one species, and the diversity within this one species. So if we were to sort of summarize what phylogeography does here, this is a summary of the steps usually incorporated into comprehensive comparative phylogeographic studies. So we take abiotic information, like the geologic history, the climatic history, the topologic history, things like that, or topographic, excuse me. And then we combine that with the biotic information, the ecological and environmental requirements of that species, the physiological characteristics, dispersal characteristics, and things like that. We combine that to do a phylogeographic study, and from that we can get evidence of biogeographic processes at the species level. And so this is things like vicariance, dispersal, diversification, and speciation, like these things that we've been talking about. But usually when we're talking about these things, we're not talking about it at the species level. It's kind of hard to tease those things out. That's why phylogeography, those methods become really useful, because we can now employ these things at the species level. and sort of merge, like I was talking about earlier, get a much more comprehensive view going from the macro scale all the way to the micro scale. And we can do that for multiple species and do things like species co-distributions and look at, migration patterns and even things like ecological interactions over time and looking back in time. So it's really, really powerful in that way. And ultimately, we can do comparative phylogeographic studies where we can look at shared biogeographic processes at the community level by combining some of these things and look at allopatric fragmentation, divergence patterns, hybridization, and genetic discontinuities. And so we're basically going, from the small scale species level to the broader scale here. So that's what's so great about phylogeography. And it really just represents an evolution of our understanding of these things, going from center origin theory and pan-biogeography to vicariance biogeography to phylogeography. Is sort of if you think back to that beginning when I was talking about sort of the merging of some of these branches, it's basically incorporating ecological biogeography into historical biogeography. So that's what has allowed us to really gain some much better insights into these things. Okay, so I want to sort of quickly wrap up with an overview of how we can zoom out some of this information and get some large-scale, better pictures of really in-depth and in time and spatial scales of evolutionary histories. So one thing that's become clear as we started using these new methods is that our previous understanding of these things is probably incomplete. So a number of phylogeographic analyses have provided evidence that transoceanic dispersal probably played a much greater role in the biogeographic histories of the southern hemisphere than has been predicted from vicariance models. So, if we think back to a lot of the things we talked about in terms of the breakup of Gondwana or Pangaea even, and how that probably explains, how we assume that explained a lot of species distributions, we've had to rethink some of that as we get these new tools and allow us to analyze things in new ways. And we're finding evidence of transoceanic dispersal in lots of different groups. So here we're just seeing some examples of all of these different groups and scenarios in which they've probably crossed oceans, in which would be an occurrence of transoceanic dispersal. So it's much more common than what we probably thought, and is why we've had to move away from some of these vicariance models. Not that vicariance doesn't probably explain tons of the relationships that we see, because it does, but we can't just, it's a little bit too simplistic to account for all of the nuance that we see in some of these distributions. And we see different patterns, whether we're looking at plants, animals, or plants. So here's historical dispersal events between major land masses of Gondwana, indicating differing patterns between animals, which is in A, and plants, which are in B. And the overall patterns look pretty similar, but basically the thickness and size of the arrows indicates the frequency, and that's where we start to see some differences. Sort of specifically, if we're looking at the patterns from Australia to Antarctica to South America, those frequencies are a little bit different for animals and for plants, and that would make sense because animals and plants rely on different dispersal mechanisms. If you think back to when we talked about some of that, as far as whether it's active or passive, or the specific mechanisms can be different, and that's going to impact how things are moving around. So we see those different patterns between animals and plants, specifically in these transoceanic dispersals across things like Antarctica to northern South America and Australia, and New Zealand and Australia and South America. If we look at Laurasia, which remember was just the sort of the northern part of that Pangaea, areas of endemism and movements between those areas, we see some really interesting patterns here too. So you're sort of seeing there's basically high endemism in Western North America and Eastern North America, in Eastern Europe, or well, I guess just Europe as a whole, and then in Eastern Asia, and those correspond to these um, acronyms here. And so basically what's being indicated here is movement between those areas. And some of this you would expect, you would expect movement to be, this is in relative terms, so just kind of ignore the numbers and look at the thickness of the lines here, and indicates relative movement. And yeah, you would expect movement from Western North America to Eastern North America. But there's also some more unique stuff going on here, like, in Eastern Asia and Eastern North America has some of the highest dispersal rates and connections. And so we need to think about, why would that be? How would this be connected to areas, any of these areas over here? And we see it, it just sort of looking at different ways of looking at the same thing here. But I think it makes more sense if we were to actually go back in time and reconstruct the geology of this. So this is a reconstructed paleogeography of the Northern Hemisphere during the early Paleogene, which is going back I think 60 million years. And so we can see some of these landform connections here that we may not have expected to see. And something else you see is this, remember when we talked about the geologic history, there was this transoceanic seaway basically across the shallow sea that formed across North America that actually separated this. So that would explain why you might have higher rates of connections in between areas of endemism between Eurasia and Eastern North America, where it may have been easier to get there, at least if you're a terrestrial organism going across land masses. So it's something it's kind of interesting looking at the specific histories, and we can tease some of this out when we start to look at areas of endemism and look at their evolutionary histories. I think it might be a little bit easier to see this way. So looking the same thing here, just in a different way, reconstructions of the paleogeography going back about 60 million years. You can see there's these connections, lots of connections, but what we know about this time period is that continents are actively drifting away from one another, and over time those connections decrease. There are still some, obviously, but those connections have decreased. And one other thing, a couple other things I want to note here before we finish up. One is that what we know is that following glaciation events, things don't move around the way that you would expect them to. We saw that when we looked at the Pleistocene, and we're seeing it more here in these organisms, again, looking at the late Pleistocene. So after the glaciers recede, we don't necessarily see northward movement, consistent northern movement. Different organisms move around in different ways. And so we're learning more and more about that, and it's shedding new information about how dispersal can influence the evolutionary histories, because basically organisms are moving around in much different ways than we expected, and much more frequently than we expected. Another example, just one quick last example here, the geographic range evolution of neotropical bird lineages demonstrating asynchronous divergence times across major barriers. So basically you're seeing the timeline for when dispersal events occurred across these major barriers, these major rivers and mountain areas. And what's interesting is that it's a little bit hard to see here. I should have blown this up bigger, this image. But the blue dots represent canopy bird species, or upper canopy bird species, and the pink dots represent lower canopy bird species. And basically the dispersal events for canopy species, upper canopy species in these blue dots happen much more recently than the pink dots, which are corresponding to lower canopy. So basically, the upper canopy birds tend to be better dispersers than the lower canopy birds, which is an interesting sort of ecological characteristic that might be influencing dispersal ability, or related in some way to dispersal ability. Okay, so I'm going to finish this up, kind of going on for a little bit there. There's one more reading for you to do here about another flower, an orchid species, and its specific dispersal mechanisms, and how that might be related to some biogeographic patterns. So keep reading through that. And then I just want to quickly summarize some of the main points here. So basically, the center origin theory and pan-biogeography represented incomplete understandings of biogeographic histories. That led to the development of vicariant biogeography, which was a step forward, but as we've seen is often too simplistic to accurately predict species evolutionary trajectories, usually going backwards evolutionary histories, which has led to the development of phylogeography, which is a bridge between the macroevolution and microevolution that allows for more robust understandings of historical biogeography. Okay, so that's sort of summarizing the main points of this module. So I will finish up there. As usual, keep working your way through the module, and I will see you next week in next week's videos.